Alright, switching presenters here. So the next question the authors wanted to ask was, does coumarin promote endothelial cell activation? Now, by endothelial cell activation, they're referring to the pro-inflammatory responses produced by the cell, such as the recruitment of leukocytes and monocytes.
The first thing the authors did to see if coumarin did indeed promote endothelial cell activation was to silence coumarin. They did this via shRNA. This is a common method used to silence specific genes within a genome. As a control, they used non-silenced cells. Via Western blot, they confirmed that coumarin was indeed downregulated when compared to cells that were not silenced and beta-actin, a commonly conserved protein. This can be seen in the top figure. They further confirmed this through an ELISA, which is a common method used to determine protein production via spectroscopy. In this case, it can be seen that the control cells had a much higher production of coumarin, and the silent cells had lower production when compared to beta-actin, as shown in the lower figure. Now that they were able to confirm that coumarin was indeed silenced, the next step the authors took was to look at the levels of TNF-alpha and ICAM1 secreted. After exposure to TP92, silent cells were seen to have a lower production of both TNF-alpha and ICAM1, as seen in the shown figure when they were compared to the non-silenced cells. This was again confirmed via an ELISA. After confirming that coumarin did indeed cause secretion of both TNF-alpha and ICAM1, the next thing the authors did was look at the dose-dependent production of both immune proteins. They found that as the amount of coumarin introduced to the cells increased, the amount of both TNF-alpha and ICAM1 also increased when compared to control cells not exposed to coumarin. This can be seen in the graph below showing dose-dependent production of the immune proteins. This was again confirmed via ELISA. So, where does this all fit in the overall aspect of the paper? Well, as coumarin has been seen to be activated by the presence of TP92 in the previous experiment, the next logical step was to see if coumarin itself can activate any immune responses. Through silencing the gene that produces coumarin, it was shown that the endothelial cells do indeed promote the immune-associated proteins cytokine TNF-alpha and glycoprotein ICAM1. These proteins are associated with inflammation of endothelial cells. Thus, exposure to TP92 does indeed cause inflammation through the coumarin pathway. Logically, the next step would be to further investigate this pathway. This does indeed support the hypothesis, as they were able to show that the coumarin pathway does produce an immune response. The next question the authors wanted to ask involved looking at receptors for coumarin. Through literature searches, the authors settled on 3 receptor proteins located on endothelial cells that may have a role in activation of said cells. They settled on CMKLR1, CCRL2, and GPR1. Pictured below is an image of said receptors. Out of all known receptors to interact with coumarin, chemokine-like receptor 1, or CMKLR1, was the most well studied. In a completely similar method as used before, CMKLR1, CCRL2, and GPR1 were silenced in HUVEC cells via short pin RNA. Like the other experiments, they confirmed the silencing of each receptor protein via Western blots. This can be seen above for each receptor. This was again compared to beta-actin and a non-silenced control. As seen in the graphs, when compared to beta-actin, the silenced cells produce the receptor proteins in much lower amounts when compared to the control cells. After confirming that each receptor type was silenced, the authors exposed each silenced cell to coumarin and monitored the production of both TNF-alpha and ICAM1. It should be noted that the paper does not specifically say the amount of coumarin used in this specific experiment. However, if similar to other experiments, the concentration of coumarin should be about 310 picograms per milliliter, and the duration should be around 24 hours. The determination of the amount of TNF-alpha and ICAM1 was also done in a different methodology. The authors used qRT-PCR instead of inducing protein production. This methodology first takes the amount of messenger RNA and converts it to DNA in a thermal cycler. While this conversion is happening, the reaction is monitored in real time. The delta-delta CT method was used to calculate the amount of an initial RNA in each cell type, with beta-actin acting as a control. Results were then expressed as a mean plus or minus standard error. This is used to determine the standard deviation. Triplicate replications of the experiment were then done and analyzed using a student's t-test. This test is commonly used to determine if data is significantly different from another set when the statistic is expected to follow normal distribution and if the scaling term is unknown. In both tests, TNF-alpha and ICAM1 production were greatly reduced in the CMKLR1 silent cells, while the other receptor proteins maintained very similar production rates when compared to the control. As chimerin has been seen to be activated by the presence of TP92 and produce immune proteins in the previous experiments, the next step the authors took was to identify the specific receptor or receptors responsible for the activation of the immune response. Through silencing 3 different receptors, they hoped to further elucidate the path TP92 uses to activate the immune response that causes aortitis. It was shown that the endothelial cell receptor responsible for activation of said immune proteins is exclusively CMKLR1. This somewhat goes against the previous literature, as chimerin has been shown to bind to the 2 other receptors. Though they may not be responsible for immune system activation, they may have some sort of unknown role. CCRL2 has been previously reported to bind chimerin and increase concentration of bioactive chimerin, thus promoting CMKLR1 activation. GPR1 has also been reported to have similar characteristics to CMKLR1 and have a greater affinity for it than CMKLR1. The next step the researchers took involves looking at another type of immune damage caused by the chimerin CMKLR1 system. Now, as all 3 receptors are also present on macrophages themselves, the next question the authors wanted to ask was, which receptor on derived macrophages is involved with migration to HUVECs expressing chimerin? Monocyte recruitment into inflammation sites plays a very important role in vessel inflammation onset. Previous research has demonstrated that chimerin can promote leukocyte chemotaxis through CMKLR1 in expressed cells. In this figure, the authors wanted to confirm that TP92-induced chimerin could promote migration of macrophages. As done before, in this panel, the authors wanted to individually silence each receptor for chimerin in an effort to deduce which chemokine-like receptor was responsible for interacting with chimerin. They used shRNA tailored to each receptor to silence each receptor and confirmed it via Western blot using beta-actin as a comparison and control cells not silenced. As shown above in the Western blot, they did indeed silence their specific receptors, and when compared to the beta-actin in the control cells, they produced larger amounts of each receptor as shown in the graph below. Here the authors did a transwell assay with each TP1-derived macrophage that had each receptor silenced individually. This assay is a commonly used test to determine and study the migration response of selected cells to inducers or inhibitors. The result showed that the only set of silenced macrophages that did not respond to TP92 was the CMKLR1 silence set. Neither CCLR2 and GPR1 silencing prevented the migration of macrophages to media extracted from cells exposed to TP92, because when those were silenced, there was no impact on macrophage migration. This was analyzed via chemoattractant index, which is a common method used to determine if an indicated substance acts as a strong attractant. The higher the index, the stronger the attractant it is. So where does this fit within the overall aspect of the paper? Well, this data adds additional support to the hypothesis that TP92 causes an immune response. Macrophages are a type of white blood cell found throughout the body and are part of the innate immune system. These cells are often associated with phagocytosis of many things considered foreign to the body and causing inflammation. Through silencing of the same receptors on the macrophages, the authors were determined to find if TP92 plays a role in attracting macrophages to the afflicted area. Like the HUVEC cells, it was seen that CMKLR1 was exclusively responsible for this immune response. If given the ability to continue the experiment, we would choose to use an in vivo assay instead of continuing to work with isolated cells. There are many things that can continue to change when moving from an indirect cell test to actually stimulated in the body of an animal. Multiple systems interact with each other and can have an effect on the expression and reception of proteins. By stimulating an in vivo system with a living mouse and the TP92 protein, we hope to identify if the system outlined within the original experiments still holds up within a living body. As there is no animal model for this microorganism, we hope to elucidate any variations within the results found in this paper. We hypothesized that mice injected intravenously with the Treponema pallidum surface protein TP92 will exhibit symptoms of cardiovascular syphilis and show immune responses within their individual endothelial cells. For the experimental design, mice will be injected intravenously through the tail. Injections will consist of 2 milliliters of 1 microgram per milliliter concentration of TP92 protein and saline solution. This will occur weekly for the duration of the experiment. We will have a control set of mice where everything will be kept exactly the same except the TP92 protein will be boiled to inactivate it before injection. In total, 100 mice will be used for this experiment, 50 experimental and 50 control. Mice will be monitored over the course of 24 months. With every 3 months, 5 random mice will be sacrificed from both the experimental and control, for a total of 10 mice sacrificed each time. The endothelial cells from the site of injection, the aorta, and the aortic valve will be harvested right after sacrifice. Aortic tissues will then be observed and compared to the control animals. The elasticity of the aortic musculature and the amount of cellular death will be monitored. As for the endothelial cell samples, cells will be cultured for 24 hours and the supernate will be examined using an ELISA kit similar to the methodology done in the experiment above. Levels of chimerin, TNFK-alpha, and ICAM1 will be observed using ELISA. With this, we hope to observe if there are any differences in the way TP92 or the chimerin system functions when exposed to an in vivo research model. As well, this long-term study will provide invaluable information regarding the progress of the disease within the aortic and endothelial tissue. And with that, I'd like to thank you all for sticking with us and paying attention to our research project. We learned a lot with this project, and we hope you learned something too. Thank you very much.